Good afternoon. Energy is renewed. We are now ready to make the most of what the day has to offer. And what it has to offer right now to us is our 4 first-handed accounts of innovation in Biospain for drug discovery and development. Moderating the panel, is Gijs Jochems, General Manager of Promega Biotech Ibérica, and sharing their knowledge with him, please welcome Antonio Quiles, High Content Screening Manager at A4Cell, Susana Pascual, Chief Scientific Officer of ICANN Biotech, Maria Jurado Puello, Application Specialist and Technical Support Manager at Promega Biotech Ibérica, and José Julio Martín Slas, a European ambassador and consultant with Science Meant Lab Consulting. This panel is possible thanks to Promega in collaboration with SLAS. So we are all ears. Thank you.
Okay, good afternoon. It's a challenge always, this first session just after lunch, and especially in a place like Pamplona. But we'll try to do the best to keep the energy in the room. Okay. High throughput screening. I think we're at a point where there's a lot of change taking place, a lot of challenges in screening, how we can reduce attrition, how can we optimize the results from very oftentimes unique samples and get the most out of this, multiplexing, multiple results from one single experiment. Very important. More and more we look for physiologically relevant data. Models that are as close as possible to the disease that we're trying to target, hence the interest to go after 3D models, three-dimensional cultures, and of course multicellular organisms like zebrafish that we will hear about. And of course we should be take a very close look also at what information technologies can bring to us in terms of data-driven science, how we can maximize the outcomes of all the experiments and try to predict outcomes, try to avoid unnecessary experiments and associated costs, of course, cloud-based science, everything that has to do with this. And then, well, we heard a lot this morning about COVID-19 and the pandemic and all the research that has gone into the virus, but also very impressive, the second session this morning, talking about high throughput. So the immense work that has gone into the production of the vaccines. And of course, COVID-19 as such has had also an impact on high throughput screening platforms. And I think all of this is something that with the panel that we have today, we can address. The intention is to have 4 short presentations of about 10 minutes each, and at the end we will have like a 15-20 minute roundtable discussion, and of course we will invite also the people in the audience to pose their questions. So without further ado, then I will introduce Antonio Quiles, first speaker. He's High Content Screening Manager at A4Cell, and he will talk about the smart chips and the cell sensing and the dynamic film about self-healing. That sounds very appealing.
Thank you very much.
Floor is yours.
Okay, so can you see the presentation? I don't know. Okay, there we go. So good afternoon to everyone. I'm very happy to be here, and today I would like to talk about cell sensing and offer this concept we are working in at A4Cell to offer this dynamic film about self-healing. So basically, what we do at A4Cell is trying to answer this question about this challenge that there's today at the pharma industry, okay, and specifically in the early drug discovery process. So many of you probably know that this process aims to identifying and validating new drugs and targets. So for this, it's very important to maintain the cell viability and working as close as possible to the physiological conditions. So for this, we'll improve the data output and we'll have more precise assays. So in the end, we'll reduce the percentage of failed drugs and improve the analysis precision. What will mean that there will be a reduce in the drug spending and thus more process efficiency and accuracy. So specifically, the big challenge in live cell imaging analysis, which is a widespread technique used in this early drug discovery process, is to be able to work with living single cells and tracking them during extended periods of time and detecting several parameters at the same time. So for this, at A4Cell, we want to offer, excuse me. Okay, we want to offer a solution for this challenge that there's still in the industry, offering the SPA chip technology. So SPA chip means suspended planar array, and basically what we are producing is devices based on the use of silicon inert oxide, on which surface we can print multiple fluorescent proofs that can be deposited to provide intercellular readouts over long culture periods. So as you can see in the images here, you can see that this is an inert micro device of 3 microns wide and long. Sorry. Okay. Sorry, where we can deposit different fluorescent proofs that it's very easy to identify and where we can quantify the intensity of this signal. Then these devices are added to the cell culture, and then the cells internalize the SPA chip without being affected in any way, and they are keeping their viability and their proliferation capabilities. So what we offer is basically the new CytoCheck SPA chip detection kits as cellomic reagents which are compatible with high-content screening platforms and other image analyzers. So as I just told you, these devices are non-toxic for the cells, so because they use very low proof concentration, and they allow us to keep track of the cells over extended periods of time. At the same time, we are detecting several parameters due to the different fluorescent proofs we have on the surface of these devices. And of course, to be used in living single cells in real time, what can improve our experiments being the perfect tool for enhancing phenotypic assays. So how does SPA chip work? Basically, our silicon device has the potential to be chemically functionalized with any biomolecule that you can imagine. And due to the specific improvements in surface chemical modifications, we can apply different kinds of fluorescent proofs. In the first case of the products we have ready to commercialize, we are going to be measuring pH and calcium, but also we are able to put into the surface aptamers, proteins, antibodies, whatever you can have in mind. And at the same time, this will be compatible with other detection capabilities, such as other fluorescent proofs, other fluorescent proteins, etc. At the same time, you can locate the SPA chips due to their different effect in reflected light, digital phase contrast, and bright field imaging, etc. Okay, there we go. So also, I would like to mention a specific case study that we are really proud of, and I think it sets a milestone for us in our technological development, and is our personal record for being able to monitor a cell culture for over 1 month, being the cells completely viable and and being able to assess and quantify the fluorescence intensity. This was performed using this cell line, RP19, which is an epithelial cell type. where the proliferation is inhibited upon contact, so the cells stop proliferating, and we can keep them in a stable culture, but just refreshing the medium. Also, in this picture to the right, you can see this happy face, and this corresponds to a culture of primary hepatocytes. So these cells were also behaving the same way as any other cell type that we have tested so far. And of course, keeping their viability and their proliferating capabilities. So we have performed different tests to check the competition of our product to what's already in the market. And specifically using our pH detection kit, we have seen that when compared to other commercial pH proofs, that they affect the cells after 24 hours in culture, and they stop proliferating, and we can see that they are severely affected by this toxic compound. Our SPH chips do not affect to the cells as they behave similarly to the control. We have also performed different experiments, checking the mitochondrial membrane potential, etc., and there's no known effect today. We have also different projects ongoing to check the metabolomic and proteomic state of these cells, but so far we can ensure that there's an absence of cytotoxicity and an increase of cell viability when compared to proofs in solution. So these two first products that we have ready to launch into the market correspond to the pH and calcium single detection kits, and you can see here the calibration curves that we have to measure different pH levels from acidified conditions to neutral pH, and also the intracellular calcium levels moving on the whole physiological range of the intracellular conditions. So we have been working together with different players in the market, and specifically in the high content screening sector, for automating the imaging and the detection of these assays. And we have worked with multi-well plates and automated imaging to obtain this kind of pictures, where you can see here the SPH chip fluorescence alone, and also in combination with different stainings in HeLa cells. In the end, to automatize the whole process and being able to analyze the image automatically to obtain large amount of data that can lead to faster and more precise results. Additionally, these products can be used in flow cytometry applications, and you can see here how the different populations of cells being exposed to different intracellular pH conditions can be easily isolated, and then in the end, using flow cytometry sorting, can be plated again in different cultures for longer periods of culture time. So finally, I want to sum up this talk by introducing to the different products that we have in our pipeline. As I already mentioned, we have the free calcium and pH single detection assays that can also be measured simultaneously in the multi-detection assay, so we can have both fluorescent probes onto the same surface of the SPH chip. And we are also working for developing these reactive oxygen species, senescence, and gene expression detection assays. And of course, we're open for developing any kind of customized detection assay in collaboration with other companies and research centers. So other projects that we have and we have been developing lately is to look for new applications apart from the use in basic research. We would like to use this SPH chip in cell therapy as a carrier, where we can deliver different kinds of molecules on even whole microorganisms. Also to work inside organ-on-a-chip and microfluidic systems to look for these more physiological conditions and these 3D cultures that everyone is talking about. And of course, further use in diagnostics as we think that they can be a useful tool. So we ask you not to miss the train, of course, and join us, because we have the opportunity to do things in a different way, and we really think that these are the things that will lead us to success. So we'll be happy to have you on board. So that would be my presentation. Here's my email, and also we'll be answering questions. So if you have any question, I'll be happy to answer.
Okay.
Now
Thank you, Antonio. Yeah. I think it's a very good example of how we can, well, work inside the living cell and try to obtain as many parameters, relevant parameters as possible. Okay, let's move on, and now we're going to real animals, and we're going to talk about zebrafish. So we're very happy to have Susana Pascual here, she is the Chief Scientific Officer from Icon Biotech, representing Navarra as well here in this audience, and she will talk about zebrafish as an animal model for in vivo drug testing.
Gracias. Well, first of all, I will just do as Antonio has done, and I will stand still because for talk, for me, it's really hard to be seated. So first, I would like to say thank you for the organization to give me the opportunity to be here today talking about zebrafish as an animal model for in vivo drug testing. Let's see how this will work. So first, I will have this movie, thank you, that will show you how fast this organism can be during development. So you also have, and now I will try to point the pointer here, so I'm sorry to give you my back to you. So this is a movie, and here you have the schematic representation. This goes from the one-cell stage until a complete embryo is formed, and this just takes 24 hours. So just so you have an idea how fast the animal model, even the development, can be. Also, yeah, they also do a bit of breakdance sometimes. So now I would like to have another picture.
Takes a while.
Perfecto. Gracias. So it was to be one picture at a time, but we have already the two, it's fine. So this is what you can see. So if you open this chorion, and if you have this animal out of the chorion, this is exactly what you can see at 24 hours post-fertilization. So just so you have an idea, this is the head of the fish. Here we have the eye, the yolk, the heart will be here, and this is the fin fold. So you usually can divide, this is the anterior part, the posterior part of the, in this case we will call it an embryo. 2 days later we will call it a larvae. And here I would like to introduce you to an adult zebrafish. And this guy will reach at max between 4 to 5 centimeters, just you to have an idea how small it is. So my next slide is exactly the question, why zebrafish? And here we go. So first, as I show you just this slide before, it has a really short lifetime, has a small size. Very important is transparent. So this means that you don't need that much to see what's happening inside of this animal when you are doing your experiments. Also, if you put cells inside, you can follow, or even if you have drugs, they are labeled, you also can follow them and see for which organs or what is affecting or what's going on, just live in vivo, just using like a normal stereoscope. Other thing that is very important is the homology with humans. So the fish will share 70% of genomic homology with humans. And if we are talking about human disease or genes causing human diseases, we will have 84%. The tissues and the organs are also very comparable and is really easy to manipulate genetically, especially because the genome has been almost all sequenced for zebrafish. So, continuing with the advantage of this great animal model, What you also have is a conserved, so it was already published for several labs, a conserved drug metabolism with mice and human. That means that you can use the drugs and you can expect a similar effect. And it's easy to do this, why it's so cool to have a high throughput drug screening using this animal model is because the compounds can be absorbed by the skin or by the gills or the mouth of the fish. And if you'd like to use, for example, an adult, you can do by intravenous injection or even for gavage, like you do in mouse. And this graph here, it just really tells us how great this animal has been, because in the last years, you can see the increasing of number of publications using zebrafish as an animal model, what gives a great scientific validation of this model. So just to make a little resume of what is all the advantage of this animal, you can see here, again, the genetic similarity with humans, how good it will be to do high throughput screenings, the cost effectiveness, the less quantity of target compound, and of course the compliance with the principle of the 3 Rs. If you compare directly with the mice, so you will have a much lower average cost per assay. Genetic similarity with humans will be equivalent between both animal models. Amount of compound used will be less, and the assay time, it will also be less. So which kind of applications can we have using this model for drug screenings? So first, you can use it, for example, for drug toxicity and efficacy. So you can use like a 96 well plate. Since they are so small, you can put 1 fish per well, and you can treat with your library of drugs. You can have these kind of readouts, like embryo and fetal developmental toxicity, central nervous system toxicity, seizure liability, also for abuse, it can be used, hepatotoxicity, nephrotoxicity, and cardiotoxicity. This is very easy to use, even because you have softwares that can make the readings of all these parameters and can give you an answer like in 1 week top. You also can use, imagine that you have a library of compounds that will be that you'd like to know which one will be the best for, I don't know, treat a bacteria, for example. So what you can do, you can infect your zebrafish larvae with this bacteria, and you can use your chemical compound library. And you can even have a really fast readout. Like if your larvae, it will be infected and dead, yeah, so your compounds are not that efficient at all. If they will be cured and dead, then they can be efficient but toxic. Or if they are cured and alive, then it's the win-win situation. So it will be efficient and non-toxic. Moreover, they also are great for rapid high throughput drug discovery. So again, you can put just the eggs in this well plate. You can treat with your library of small molecules. You can have a lot of them at the same time. And you can just check for a phenotype assay. So which kind of abnormalities you will have in your larvae. Do you have like a head abnormality, like something at the level of the brain, heart? What will be the shape of the fin fold, and so on. So you really can isolate all the effects of these small molecules also like in 1 week. So now I would like you to pay attention first to the right part of this panel. So zebrafish, since I also told you that it's a great genetical model, you can really mimic human diseases using fish. So for cancer, for example, or even for rare diseases, if a mutation that is cause a disease, it's found in a patient, you can build your fish in the lab. And then, by doing that, you can treat with compounds, for example, that you have in mind, and you can make it by the efficiency of the compound for this really target disease, or also evaluate the toxicity at the same time. If you would like to be a bit faster, what you also can do for the case of cancer is you can inject cells directed from a tumor from a patient, and you can inject in the fish. And this will give you, in the time of a week, a prediction of the disease. So if these cells will migrate or they will proliferate, what is going on first, and then you can treat again with several compounds of your interest, and you can check what will be the readouts. So if this tumor will shrink, the cells will not proliferate anymore, so what will go on, and at the same time, how it will be the patient, in this case our fish, so is this toxic or not. But zebrafish is not just great for like chemical compounds test. It starts also to be used, for example, for immunotherapy in vivo. So in this case, I don't know if you remember for the first slide, this is the posterior part of the fish, this is the fin fold, and this is the circulation. So what you can see here is cells circulating. What was injected here, it's NALM-6 cells, so it's leukemia cells, and these cells present like a flag that is CD19. So what you have here is the tumor cells that are in green. I don't know if we can put the movie again working, that will be great. But this is to say that the green cells that are in circulation, and this is CAR T cells. CAR T cells is T cells that usually your immune system produced. And these ones, what happened is that, thank you, is that have been engineered in a lab. So this means that these cells will target this because of this CD19. And what you can see is like the red cells are going to the place where the green ones are, and they will kill the leukemia cells. If you don't believe me and you think it's just the red is covering it, so you can see here a stack of this movie. And as soon as 19 hours post-injection, you can see that all the green cells are gone. Other kind of experiments that you can do, and because now what is also really important is try to find kind of therapies that will attack just tumor cells, but not, for example, healthy tissues. It will be the case of nanoparticles. So in this case, we will have the cancer cells labeled in red. So this is like a melanoma tumor here. You can see the vasculature of the fish that are in green, and the nanoparticles are here labeled in white. If you have a magnification of these, you can see that the nanoparticles are just in the tumor side and not in the other tissues of the fish. This, you also have here a quantification of the cancer tissue for the accumulations of nanoparticles and not in the healthy tissue. And this brings me, so for my last slide, and I will be really brief, I hope.
Yeah. No.
No?
One back.
Okay, perfect. So, and just to introduce like what we do in Econ Biotech. So it will be my last slide. This is our workflow right now. So we are very happy because right now we have a platform that is working, and we have stable cell lines. Right now we have colorectal cancer. We are working with colorectal cancer. We have stable cell lines, and very, very important, we have a library of biopsies that are characterized already. We do also in vitro studies. So for example, if we have several drugs, we will know by in vitro. If you have 10, you can select like 5 that will work the best. And we can do it like in adherent and also spheroids, so in monolayer and 3D culture. We also do the treatments of acute toxicity for the drugs in the fish. And we have all the platform for the xenografts. So we can inject these cells in the fish, we can treat, and then we have several readouts like human genomic DNA quantification and also RNA expression, what will give you the profile of these cell lines or of these biopsies before and after the treatments. And at the end, we will really like to have all of this data armazenated in a software that could be used artificial intelligence that could be a predictive platform. So also just to tell you that we are in a way that we can also testing several drugs using our platform. So I'm not sure, but I think I'm already talking more than 10 minutes. And so if you have any doubts, have any questions, please just write to me or to Ruben Diaz. He's also here today, and this is our emails. And thank you so much for your attention.
Thank you. Thank you, Susanna. Very interesting. It's very appealing to see how we can now start screening our libraries on something that is really close to the real disease state. So that is in the end where we all end up, right? So now we will move on, and this year we are celebrating 30 years of bioluminescence in our company. And Maria Jurado, who is leading our scientific applications team, she will go over what we have been achieving over these 30 years and how we can illuminate the live cells.
Yes. So thank you, Gijs. So yeah, I'm going to try to illuminate a little bit drug discovery in live cells. So I will talk about bioluminescence. But let me start saying that at Promega we are quite concerned that it is very important to choose a relevant model to investigate in research. So you can get different responses from different cell models. So we are making a lot of efforts in trying to test our new assays that we are developing in live cells. We are trying to develop real-time experiments, trying to work at the endogenous levels, especially when we are talking about protein reporters. We are trying to focus in multiplex, meaning reading 2 or more parameters in one same well, in one same population of cells. Also working with different 3 models. We are using mammal 3 models, but we have collaborations with labs that are working with other kind of 3D models. iPSCs, or also trying to use a lot of co-cultures and bioassays. So we are concerned about this, and we are making efforts on this. And at the same time, at Promega, we've been 30 years fascinated by bioluminescence. And this is not only because of the beauty of the luminescence, but more important maybe is that it is very sensitive and it has a huge dynamic range. So we compare a luminescent assay with a fluorescence and a colorimetric assay for reading one same parameter. In this case, it's a gas-phase activation. We always see a much better sensitivity and a huge dynamic range. So this is very important. And the second reason is that bioluminescence is very compatible with high throughput. So it's very adaptable to this kind of plates, and we've got very good results using this kind of high throughput plates. So with these 2 reasons in mind, we've been working a lot with bioluminescence in the last 30 years. And in 2012, we developed a new luciferase called NanoLac. This luciferase is very, very small and very bright, and these 2 characteristics make it very, very interesting because we are now able to do much more applications than before with previous luciferases. And since it's very bright, we can also use it at endogenous levels. So when we try to use it, for example, as a tag for proteins, we can work at endogenous levels. And we can also work in live cells now because we have a substrate that is permeable to the cell membrane. So we can work this way now. So one important part of our new assays that we've developed with NanoLac are these cell health assays to work in real time. So you can work with the cells in the culture, live cells, and you can work up to 72 hours for some of our assays, or sometimes more. If we change the protocol, we have some modifications that can be used to extend that period. So you can monitor as much as you want during that period the viability or the apoptosis activation in a cell population. Also, we've explored a lot of protein reporter applications with NanoLac. There are 2 technologies for protein-protein interaction in live cells. The first one is called nanobit, which stands for NanoLook binary technology. So in nanobit, what we did was to split NanoLook in 2 parts, large bit and small bit, so 2 bits of NanoLook. And these 2 bits have very low affinity between them. So if we couple one of each to different proteins, we can have this expressed in a cell, in a leaf cell, and we can monitor in a reversible way when these two proteins are interacting. So this is driven by the proteins. So this is called nanobit. But we also have another technology called nanobread. Nanobread stands for nanolook bioluminescence resonance energy transfer. So it's similar to FRET, to the FRET concept. But here, the donor of the energy is nanolook. So again, we need to couple, in this case, it's nanolook to one protein, halo tag with a fluorophore, which is a different tag, to another protein, and we can monitor in the leaf cell when these two proteins are close enough, we will have this transfer of energy. So nanobread technology has also been applied to a different application, which is called target engagement. And we've been very prolific. We have many different assays developed for different proteins for target engagement. I will talk about it in my next slide. So target engagement means to study how a drug binds to its target protein. So we can use nanobread for that in leaf cells. And another application that we've been doing with nanolook is called HiBit, and HiBit stands for high affinity binary technology. So in this case, again, we split nanolook in two parts, large bit, and now the small part is called HiBit, and this HiBit has very high affinity to large bit. So this technology can be used to label one protein with this small tag called HiBit, and then we can detect the protein. We can detect the degradation of the protein at endogenous levels again, or when the protein is stabilized or goes out of the cell membrane, so to monitor the protein. So now I'm going to talk very briefly about these applications a little bit more. So the first one that I was mentioning was the cell health new real-time luminescent assays. So with these assays, as I was saying, you can monitor what is happening in one population of cells in real time for hours, for days. And you can use one instrument capable of doing gas control, or you can use a regular incubator and a reader, a luminometer. And this means that you will use one plate. So only one plate when you would need multiple plates with different time points in a non-real-time format. And we have this cell viability assay that is called real-time glow. We have also cytotoxicity assay. We have an apoptosis assay called real-time glow Annexin 5. And this is for the cell health assays. Now, moving to the nanobread technology and the target engagement, this is a very nice technology that we like a lot because allows researchers to study target engagement in leaf cells, so in the context of the cell and everything that is inside the cell. This is important because now with nanobread technology, we can, in first place, we can work with full-length proteins. In vitro, sometimes you need to work with only fragments. So now we have the whole protein. So how it works? In this technology, you need to label the target that you want to study with nanolook. Then you need a tracer that is a fluorophore. The tracer will bind to the target, and then we will see how the bread signal increases. Then you will add the candidate that you want to study, and the candidate will displace the tracer from its place, and you will see a decrease in the signal of bread. And then you can also study drug residence time. So you can also now add again the tracer and see how the tracer displaces again the candidate. And this is important. You can see here that there is a difference between the curve for the biochemical and the cellular experiments, and this is important. And the reasons for this are, some of the reasons are this, but there are more. So in the cell, you need to realize that there are more things that are not present in the in vitro biochemical in the tube. So for instance, you have protein complexes all around, you have activation state of the target protein. You have the plasma membrane here. You have metabolites, and also the stoichiometry. The concentration of the protein is different in the tube and in the cell. So all this explain why the results are usually different in biochemical versus the live cell target engagement. And at Promega, we've been making a lot of efforts using this technology, nanobread technology, with the RAS pathway in drug discovery. And this is, since I don't have a lot of time, I have here a link if you are interested. But let me just tell you that we've been combining NanoLook and nanobread technology together. So this is an example where we can label KRAS with large bit and also KRAS with small bit. So this is a nanobit technology that I was explaining that works to study protein-protein interactions because the interaction is driven by the proteins. So we can combine nanobit with nanobread. So we can see this way how a drug is binding to KRAS when it's dimerized in the membrane and not when it's not there. So this is just an example to show how these technologies can be combined to answer specific and difficult questions that were not possible to answer before. And if you remember that slide, another application of NanoLook was high-bit, the detection of proteins using this small tag. So let me just show you here an example of using high-bit and also nanobread and nanobit in the pathway of degradation by a PROTAC through the proteasome pathway. So we can use this technology in these 5 key stages in the degradation of the protein. So we can, for instance, use nanobread technology to study the permeability of the PROTAC and target engagement to its target. We can use also nanobread, in this case, the protein-protein interaction to detect how the proteins are interacting, in this case, the A3 ligase that needs to bind to the PROTAC and to the target protein. Or to study how the ubiquitin is binding to the target, or how the protein is binding to the proteasome or being recruited by the proteasome. And of course, just high-bit to detect the levels of the proteins and how it is degraded in the live cell. And this is my last application, so I will just say very fast.
We need to find time for Julio.
Yes. OK. So very fast. We have a huge portfolio of functional assays to study antibodies. So our assays cover a lot of different mechanisms of action that these antibodies, therapeutic antibodies or antibodies produced by a vaccine can mediate. So, FC effector activity, cytokine modulation, immune checkpoint modulation, these are a mechanism of action of these antibodies. And we have bioassays that are based on co-culture of, in this case, this is an assay in this pocket, the immune checkpoint modulation. So we have a co-culture of effector cells and target cells in one same well. And we can use this kind of assays to detect the mechanism of actions of the antibody. If you are more interested, I can speak to you later if you want. And that's all. So if you have questions, you can ask later, or I have here we have a technical service support in Spain, so it's a local technical support, and we are happy to help in any questions you may have. Okay, sorry.
Okay, thank you, Maria. That was a lot of information. But it shows the versatility of bioluminescence and, well, brightening up and lightening what can be detected within a living cell. So now finally we have Julio. We still have time, and well, Julio Martin is well, I think, very well known to everybody who is working in the field of drug discovery in Spain. And well, you're now freelance, but you've been working for a long time in GSK, and now you're also, since not too long ago, you're now ambassador for the Society of Laboratory Automation and Screening. So very pleased to have you here, and
Thanks for the invitation.
Try to wrap up everything and enlighten us for where the future is in drug screening.
Yeah, thanks for the invitation and introduction. Yeah, so the purpose of my talk is what Gijs just said, is trying to wrap up and trying to put together all the pieces of the jigsaw being early drug discovery, right, in pharmaceutical R&D. And let me start by setting the scene of pharma R&D. So pharma R&D has been living over the last, I would say, 2 decades, if not longer, in the perfect storm because of the continuous decline in the productivity in new pharmaceutical treatments. And the main reason for that is the high attrition rate. So it's the level, unacceptable level of attrition. What's the reason for that? And the reason for that are multifactorial, but let me focus on 4, simplify on 4. One is maybe the target, so the wrong selection of targets, the wrong selection of molecules, because we neglect critical properties just because we focus on the potency, on the affinity of the compounds at the target in the first assays, and we neglect the pharmaceutical properties, which are relevant for development. We don't know how to navigate the leap optimization, the molecule optimization in drug discovery. So we have a lot of data, a lot of information, but very little knowledge, or not as much knowledge as we need to make decisions. And last but not least, and this is the topic of the session today, is about assays, relevant assays. So at the end of the day, it's about predict what is going to happen in more complex scenario and making the right choices and decisions, right? So we are not doing well in biological assays when we interpret those. So there might be something that maybe we can change in our minds. The best thing, as has been mentioned here, is how we can build relevant biology. So by nature, the human mind struggles the way we can cope with the complexity. And that's the reason, because we try to reduce everything to the minimum expression, right? There's nothing wrong with doing that. What is wrong is when we try to rebuild the complexity, and there is some disconnects in this way. If you like me the analogy, it would be like if you deconstruct the Spanish potato omelette, and when you reconstruct, you get scrambled eggs with fries, right? So there is something wrong there. And that's happening. So, but now we have amazing technologies coming up, like for gene editing, CRISPR, and to culture and produce stem cells, so human natural cells from patients, from healthy individuals. So we can put together, this is opening a completely new scenario, not only for the systems, it's also for natural stimulus and natural readout. We have seen very elegant examples by Promega about how to use this, and another turn in the screw by ECAN, in which you can not only culture cells for the right phenotype, to read the right phenotype in a Petri dish or in a microtittle plate, but also animals, right? So that's really amazing. So this is taking us to another different paradigm, which is from clonal samples, off-the-cell samples. So immortalized cells, you go to the fridge, and you get the freezer, and you get the samples, and you put the samples in your assays, to more personalized samples, unique samples. So that means that you have to do more with less amount of sample. So how can you figure out the assays to get this purpose? So, and we have seen also examples for that, to do multiplexing readouts, kinetic pharmacology, single-cell analysis, live cells. So the technology SPA chip and Promega technologies allow us to approach this and to work with this kind of samples. This is they are not the only technologies, of course, out there. There are many others, right? Once you have a molecule, right, you have to navigate the optimization of that molecule. You have to build new properties, optimize the properties on top of that. And sometimes we lose track of that. So we are not sure that we are in the right track to optimize. And in many circumstances is because we are not sure if we are still engaging the target. And Maria have commented about the nanolog applications for doing that. So keeping your mode of action on the right target is a guarantee that when you go to clinical POCs, you are not going to have a failure because of lack of efficacy, just because you realize that your molecule was not acting against the target that you wanted, right? And this is not unusual, okay, to happen. We have seen examples of Promega, as I said. But the other reason for failure may be, as I said at the beginning, that we are not building the right properties on the molecules just because we are neglecting those. And these are related with fish-game properties. We need, if you want to administer the drug orally, you need soluble molecules, PK, permeability, but also safety pharmacology, you know, ADME-tox in general. Here we have seen applications from these three technologies in which we can leverage those technologies to approach this and to answer this question. Right? Assays about toxicity, the readouts of the SPA chip, pH changes, undesirable pH changes, undesirable calcium mobilization changes, etc. Microtiter plates and wells is the gold standard, right, in screening, particularly in high-throughput screening. But when you start to work with precious samples and trying to build 3D biology on those, there might be some limitations, technical limitations. So the scientists are developing new formats, right? So we have to accommodate our assays to those formats. And this implies technological challenge, particularly about how to build 3D biology, right, on these devices. But it's also about incorporating detection technologies and readouts to these new devices, right? And here we have the A4 cell again, SPA chip, in which you have a biosensor inside the cell, right? So this is easy to be read in any conventional readout or reader. Last but not least is digitalization and automation. This has been always there, you know, in the screening field. But now there is a revolution, right? And the pandemic has, accelerated this process. So artificial intelligence, and next session will be about artificial intelligence, so has come to stay. It's not a foe, it's our ally, because artificial intelligence is nothing else than canning what is in the natural intelligence, right? But there are more challenges here. So now we start to see the conceptualization of what is called smart cloud labs. So can you imagine a closed-loop laboratory unattended in which you put your protocol and you get your readout, and then you forget about it? So this is starting to happen. Either it's a hype or a hope, we don't know, but it's worth keeping an eye on that, because the way we are going to design the assays will have to be transformed. okay, to accommodate these new ways of working. As I say here, there might be a paradigm shift, you know, in the discovery lab, from life sciences to information lab. Information, data sciences, for me, is the key competitive advantage in discovery. It's more than the way you run the assays. So I'm going to wrap up by these take-home messages. So better disease-relevant models, target engagement to make sure that you are on the right track, digging deeper into pharmacology, do more with your samples, you have less samples. Early assessment of developability, and in the data and chips, the technology, information might displace wet experimentation. Think of smart labs, cloud labs, and integrate bioengineering devices. So maybe we have time for some.
Yeah, we do.
I'll stop here.
Okay. Thank you, Julio. I don't know if this is working. Yes, it's working. That was a great wrap-up. It clearly pulled everything together, and it was also a look into the future of what the data-driven science can bring to the screening platforms. One thing you mentioned briefly, but we haven't really spoken about it, a bit of a link to what we have been hearing this morning as well. What has been the impact of this whole COVID-19 pandemic on high-throughput screening and pharma in that sense? Do you have something you can comment on that?
I mean, not being able to go to the lab to run the experiments, that has been tremendously disruptive everywhere. So the technologies around running labs and experiments and high-throughput screening in an attended mode was there for the very beginning. But the driver at that moment was more about efficiency, right? Decrease the manpower and get a better return on this investment, you know? But now it's different. Now it's about more, the driver is more quality and free up time of people to think about the things that are really relevant and only the human mind can do, and free up some of this time. That's for one reason. And the other is quality. If you are able to develop an assay, a protocol, robust enough, informative enough to be run in a closed-loop setting, unattended, I mean, that means that everything is going to be more reliable, your decisions are going to be more reliable and will be smarter. So I think that it can be, the pandemic has been, for me, a catalyst of this transformation, that it was there, it was incubated, but now it's going to happen.
So in the end, since we need to optimize and become more efficient, so what we've been seeing, the possibility to do multiplexing and get as much data out of your experiments, and especially as close to the disease model that you want to target, and in
now we have very
conditions.
precious samples. I mean, it's a shame that, I mean, in the past, the high-throughput screening was just one sample, one data.
Yeah.
I mean, now that's a waste of resource. I mean, if you get samples from patients and you run stem cells and, and you engineer a biosensor inside, I mean, you have to get most of it. And this will be a change as well.
Yeah. I'm not sure if there is someone in the audience who would like to ask a question to the panel here. There's probably time for one question. If there is no question, then well, there's a lot of things that we can discuss, but to talk a bit more about what this multiplexing possibility. So, Antonio, having heard the other talks, so how do you see maybe your SPA chips? How could they be working in a conjunction maybe with some of these other technologies? How do you see that?
Yeah, I think that's the main point, as Julio mentioned, the possibility to have different parameters and tons of them, and then to be able to apply artificial intelligence, that's going to give us a lot of results that a single person in the lab is not going to be able to analyze in a unique way. We can find different patterns of relationships between molecules, etc. And specifically with our technology, what we think it's going to also be important to compare to other platforms is that we would be able to mix different reactions. So, for example, now if you're running a RNA sequencing experiment, you only read the RNA. But if you can have here a measurement of gene expression and protein at the same protein levels at the same time with antibodies, whatever, that's another way of finding relationships, and that represents more accurately the physiological conditions of what's happening in the cell. And not only the cell, but also to be able to check relationships between cells and in the end represent the native tissue.
Okay. So thank you very much. I think we are on time, and we need to make space and room for the next session. So thanks to all of you. I think it was a very enlightening session, and well, also thanks to our co-sponsors, the SLAS, and then we leave it here. Okay. Thank you very much.
Thank you.
Thank you very much. Now we are going to follow and we are going to go to the next subject that we just need a few minutes just to change and clean the microphones, and we will be back soon, very, very soon.